So, without further ado, I'm Heidi Swanson, for those of you who don't know me, from Waterloo, and I would like you to welcome Tom Binder.
Okay, thank you. Good morning. I'd just like to start by thanking the organizing committee for inviting me to give this talk this morning. And I'd also like to pause just to acknowledge my co-authors. There are many. And also the funding sources that fund both my research and my travel to this meeting. The foghorns are going here. As Andrew mentioned yesterday, I was asked by the committee to give kind of a synthesis talk on a couple of chapters that I'm involved writing for the upcoming Lake Charr book that will be published as part of the Springer Fish and Fisheries series. The two chapters that I'm involved with are the Habitat chapter, which is led by Ellen Marsden, and then the Movement and Behavior chapter, which I'm taking the lead on. Now, obviously, you can imagine there's far more in those two chapters than can be presented in one 30-minute time slot, so I'll be focusing today on three specific aspects: adult foraging behavior, homing and navigation, and then spawning behavior. And my approach in the talk will be to kind of highlight some of the recent observations that I feel are challenging and expanding current conceptual models of Lake Charr behavior and habitat use. And then our kind of ultimate goals here are to propose kind of updated conceptual models that can serve as hypotheses on which to base future research. So with no further ado, I'm just going to jump right into adult foraging habitat. And while I was reviewing the literature on this topic, it was really quite amazing to me just the wide variety of lakes that Lake Charr are found in, you know, from the smallest, tiniest lakes in the Canadian Shield to the largest lakes in the world. But there was a very common theme that you tend to see across these studies, and that is that outside of the spawning period, Lake Charr look for two things. They look to stay cool, and they look for the highest quality food that's available to them. So over the next few slides, I'm just going to present a few different observations that kind of form the basis of the conceptual model that I'm going to propose. Starting first, this is something you see quite often, this idea of habitat squeeze. This happens mostly in temperate, small inland lakes, and during the summer you get a thermocline, and as the summer progresses, that thermocline pushes deeper and deeper into water. At the same time, because of that thermostratification, you don't get a lot of mixing, and the bottom layers become hypoxic. So the fish, as the summer progresses, get kind of squeezed into this ever shrinking layer of habitat that's kind of bound on top, and they're trapped below surface waters by increasing temperatures and bottom waters by this low oxygen concentrations. And the general consensus is that temperatures above 15 degrees Celsius are not tolerable by Lake Charr, and they prefer temperatures that are less than 10 degrees Celsius. And then the kind of lower limit for oxygen is about 6 milligrams per liter. So you can see here that by the end of the summer, the amount of habitat that's available to the Lake Charr is a lot less than is available in the entire lake and during other periods of the year. Another feature of a lot of these small lakes is they lack pelagic fish prey, which are kind of the preferred forage for adult Lake Charr. And this often means that the highest quality of food is available in the littoral zone, which of course ends up being too warm in the summer. So what you tend to see in these systems is this littoral pelagic coupling. And basically that's just a seasonal shift in habitat use between near shore feeding in the cool months on more high quality food like fish, benthic invertebrates, and aquatic insects, to kind of offshore feeding during the summer months on less high quality food like mysis and zooplankton. Now obviously the shift between these habitats is driven primarily by temperature, but things like prey availability and even lake morphology are also important. Now, one of the interesting observations that have come out in a few different papers is the idea that lake char will make frequent short duration forays into this littoral zone during the summer to feed on these higher quality foods, despite the fact that temperatures are well above what are considered tolerable. Now, these forays are usually very short, less than 15 minutes, and then the fish move immediately back into the cooler water to kind of digest. But ultimately, what it looks like is that habitat use in lake char seems to be a trade-off between the thermal costs and the foraging benefits of occupying different habitats. Another really good example of how prey availability seems to affect movements between habitats is this idea of, or observation, I should say, of partial anadromy in some of these northern low productivity lakes. And I'm not going to talk too much about this because there's actually a talk on Thursday that's going to specifically address this behavior. But I believe the first kind of real published observations of this was done by Heidi Swanson et al. In 2010. And they described approximately a quarter of the lake char in 4 lakes making annual migrations to coastal brackish marine habitats. So I'm not going to talk any more about that. I'll leave it, hopefully people will see the talk on Thursday. Now, unfortunately, I probably could have spent this whole talk talking about foraging habitat because there's a lot of cool stuff out there. But hopefully you guys get this sense of this trade-off between temperature and prey availability. And I'll just move ahead and kind of present this model that we're proposing. The first question is this a low productivity system with access to the marine system? If the answer is yes, then you would predict to see this partial migration into the marine ecosystem. But most of the lakes that lake char reside in do not have access to the marine system. So you can move on down here to the next question, which is, are there offshore abundant prey resources in terms of fish anyway? If the answer is yes, then you would predict to see more offshore habitat use, preferring pelagic habitat when it's available, when the fish are there, and if not, using benthic habitats. And anyone who saw Matt Corniss's talk, he talked a little bit about how lake trout in the Great Lakes seem to be switching from pelagic to benthic feeding because of the loss of these pelagic prey fish. In systems where the offshore prey fish are not abundant, you tend to see more diverse habitat use, with the relative use of these habitats being driven mainly by the thermal conditions and prey availability at any given time of the year. So that's kind of where I'll leave things with the foraging habitat model, and we'll move on to homing and navigation. So lake char tends to be very specific in their habitat use, and they tend to show repeated use of the same habitats year after year. This is data from a telemetry study that we've done in Lake Huron using positional telemetry. What this system gives you is positions that are accurate to about 5 or 10 meters every few minutes for every fish in the array. So our array covered an area of about 25 square kilometers in northern Lake Huron. And what this system is really good at doing is mapping habitat use. So using this system, we discovered 6 distinct spawning locations spread out over 5 different reefs. And those are shown here on the right. This is basically just a habitat map where the color of each little square, these are 20-meter squares, are the color-coded based on the amount of activity that occurred in those areas. And there's 2 patterns that come across hopefully really clearly here. The first is that the char are using a very small portion of the available habitat. Depending on which reef you're looking at, between 48 and 84% of all activity occurred on only 10% of the available habitat. And then the second obvious pattern is the consistent use of the same habitats year after year. And if you do correlations from year to year, you see correlation coefficients ranging from about 0.44 to 0.95, depending on reef and year. So it's pretty clear that lake char are homing to the same sites year after year, but the precise mechanisms are not known for how they actually navigate to those sites. It's been widely hypothesized that there's natal homing in lake char, but as yet there's no actual direct evidence of natal homing, at least none that I've seen. There are lots of indirect observations, and in my opinion the most compelling is work that was done by Bronte et al. In 2002, where they stocked eggs onto Devil's Island Shoal here in Lake Superior, and then so they began stocking in 1981, and I think stocking went all the way through to 1995. And then they surveyed each spawning season for unclipped or wild spawners. And what they saw is that the number of unclipped spawners on the site remained low until about 7 years after they first started stocking the eggs. And that's exactly what you would expect to see if you assume that the spawners that are being caught are coming from those eggs that were stocked onto the spawning grounds. So I think this is pretty compelling evidence that there is probably natal homing in Lake Charr, and that would be consistent with what you see in most other salmonines. One thing that, a little caveat to that is that we also know that hatchery-reared fish have high site fidelity to spawning locations. So obviously natal homing is not the only mechanism by which Lake Charr finds spawning sites. It's been proposed that chemical communication may play an important role. And up until recently, the prevailing hypothesis has been that adults are attracted to odors that are released in fry feces. But one of the issues with that is that in Lake Charr, the fry leave the spawning sites soon after absorbing their yolk sacs. So for those cues to act as a navigation cue, they have to persist in the environment for a long time, several months, before the adults are returning to the spawning sites. So this hypothesis was tested by Buckner et al., who's a graduate student from MSU who did this work up at the Hammond Bay Biological Station where I work. And they did chemical assays on the 16 putative bile acid that are released by Lake Charr fry. And in the assay, they found that only 3 acids were released, and all 3 of those degraded significantly by about 2 weeks after fry removal, and by about 4 weeks, they were below detectable thresholds by Lake Charr. So I think this is a really clear evidence that these bile acids that are released in the feces of fry are probably not acting as navigation cues for the adults, despite the fact that they did find that adult males like Foster were attracted to fresh fry feces. But when they did the assay with the residual fry feces, they found no attraction. So clearly these things are kind of dissipating and gone by the time spawning occurs. I think there's kind of mounting evidence, though, that that conspecific chemical cues are playing a role in navigation. This is more work that was done by Tyler in our field station. He used again, a behavioral bioassay and an experimental stream where he had paired reefs, and he applied various combinations of odors from males, females, and juveniles. Those were age 4 fish. And he looked at the relative attractiveness of the reefs depending on the combination that was there. And he assayed the behavior of both mature males and mature females. What he found is that the females were showed a high preference for odors from males. They also preferred the odors of juveniles over the odors of females, but the response to the male odor was by far the greatest. Somewhat interesting, they also showed that males preferred odors from other males, and that they also had an equal response to juvenile odors as for males, and both of those were much greater than the response to females. Now, on the surface, that may seem kind of counterintuitive. Why are the males attracted to other males? But when you think about the mating system of Lake Charr and the fact that the males arrive on the reefs several weeks in advance of the females, and the females actually spend little time there, the male cue is probably a more reliable and consistent cue indicating the presence of habitat than the female. So I think we still have a lot to go on that, but we seem to be making some strides. When I was reading this, it really made me think that the system in Lake Charr seems to be very similar to what they're showing in stream-dwelling salmonines. So I've kind of hijacked this conceptual model from Breton Hinch, which they called the hierarchical navigation hypothesis. This hypothesis states that kind of olfactory cues reign supreme, and fish will primarily use imprinted cues that they acquire during early development. But when those are absent, they'll turn to conspecific cues. And we're seeing evidence, I think, of both of these things happening in Lake Charr. If there aren't any olfactory cues, then they turn to kind of, as a backup to non-olfactory environmental cues, things like sounds that are made by conspecifics, environmental noise from things like waves crashing on reefs or shorelines, lake bathymetry, stuff like that. So I think that this stream dwelling or stream spawning conceptual model probably fits the Arctic char model fairly well, but I just want to add one potential, more of a question, and that is there a potential role for learning and memory? When you consider that this model is primarily based off of semoparous species, and lake char live a long time and can spawn numerous times throughout their lifetime, it's conceivable that there may be an advantage to, kind of seeking out uncharted territory, trying out new things, looking for sites that may be better than the ones that you came from, and also an ability to kind of adapt to changes in the environment and change your spawning habits. So I think that's more of a question than that there's actually evidence. I mean, if you consider the work by Gunn that showed if you cover up their normal spawning sites, they will move to other spawning sites. There is, I guess, some evidence that they are adaptive at least, but it would be interesting to know if they change their behavior over time and things like that. So the last thing I want to talk about is spawning behavior. And I would make the argument that we actually know very little about the spawning behavior of lake char. And I think a lot of this comes from the fact that they spawn in places that are hard to study and at times of year when it's difficult to work, especially if you live in Michigan and it's during hunting season. It's really hard to get people to get out there. So most of what we know about lake char spawning comes from basically just a handful of mostly above surface observations of spawning in rather shallow spawning sites, some of them less than a meter. Nonetheless, we do have a conceptual model which states that lake char spawn primarily in lakes and they don't build nests, unlike other salmonines. But there is some evidence that males may clean the sites prior to spawning. We don't know if that's a purposeful act or just an incidental occurrence based on the fact that they're there for so long before the spawning period. It's very well established that males arrive first on the spawning grounds and they're present on the spawning grounds in much higher numbers than the females. And also that spawning occurs almost exclusively at night. And then during the day, the fish tend to move off into deeper water. Now the spawning act itself occurs in groups containing one female and anywhere between 1 and about 9 males. And the interesting observation that you see over and over again is a report of no apparent mate selection or intersexual aggression between males, competition. And that's a curious observation because it's very unlike what you see in most other salmonine species. And also it's kind of contrary to evolutionary theory when you consider the huge amount of investment that goes into these nice large eggs and this highly skewed sex ratio on the spawning grounds towards males. It really makes no sense that there's no mate selection or competition. And I think that's one area where we're starting to make some ground in recent years, where different observations are suggesting that the mating system is probably more complicated than was previously thought. And we're seeing evidence that there may be competition, like kind of novel forms of competition and mate selection occurring, and that these behaviors may be occurring independent of the actual spawning act itself, where we've kind of focused all of our attention. So this started with a paper by Steve et al. In 2008, who used underwater video to get really detailed descriptions of lake char spawning behavior. And they described what they called an itinerant spawning strategy, where instead of building nests and spawning in these redds, the females travel over the spawning grounds and periodically drop into the substrate and release eggs and then continue, and they do this several times. And he described this behavior as having 3 distinct stages: a traveling stage, so this is where the female leads the group around the spawning, then they stop and sink into the substrate, and then they release gametes, and that looks very much like you'd see in other species. So this is just a quick video showing what that looks like for anyone who hasn't seen it. This actual video is coming from Drummond Island. This is a video that we took, but it looks exactly as Steve describes here. So the female is right there. I don't think actual gametes were released in this sinking incident, but so the female begins leading the group. She's now on the outside there. Sorry for the shaky hand. She leads the group around, so this is the traveling phase. There's the sinking phase, and there's the gamete release. So what the authors of that study hypothesized is that the switch from kind of red billing to this itinerant strategy caused the evolution of a novel method of competition amongst the males, where being aggressive towards one another was kind of disadvantageous because it took you away from the female. So instead of kind of aggressively fighting with one another, the males compete for space beside the female. And when you watch many hours of this video, you can see that they really jockey for that space on either side of the female, often nudging each other out. Sometimes you'll see an open side and the male will try to get in there and push the other male off. So it seems that we have kind of a novel form of competition occurring in this species that hasn't been described before. And that may have been, the blinders may have been on because we're kind of comparing it to other salmonine species. But in addition to this kind of novel competition, we're also seeing possible evidence that there may be mate selection occurring, and this mate selection might be happening before the fish move on to the spawning grounds and begin this spawning behaviors that have been described so far. And this started with work by Mir et al. In Great Bear Lake, and they described a behavior that they called finning. And finning was described as being groups of 2 or more fish kind of hovering just below the surface of the water or slowly moving with their fins erect and breaking the water. And what these guys hypothesized is that this was a form of display courtship, and they weren't able to distinguish males from females in this case, but the assumption was that these were males trying to court the females in some sort of display type behavior. And we saw a similar and probably analogous behavior occurring at Drummond Island, which we called hovering. We got a video here. So again, we see this is a female. This is a female, and these are males. The female is basically holding stationary. The reason they're moving is because of the waves. But here you see the males are really rubbing against her, nipping at her fins, quivering, things like that. And again, presumably this is some sort of courtship behavior that's happening, trying to induce the female to begin her spawning behaviors. So I mean, I think we're just getting a glimpse of what's going on here. But I think the observations are suggesting that there's more going on than we thought, and that these things might be happening either temporally or spatially separated from the actual spawning event itself. So again, I'll say I think we still have a lot of work to do here, but I think we are starting to make some headway. Where I think we've added to the conceptual model is the idea that there is probably some sort of mate selection going on, and that this might be happening independent of the spawning reefs. And you know, one hypothesis I have is that the reason the fish move offshore during the day is not because they're afraid of light, which is kind of the current hypothesis, but because these behaviors may be happening in deeper water, and especially when you think of shallow water shore sites where the fish are spawning in a meter or less, these fish might have to move off of the spawning grounds in order to conduct these behaviors. So I think we've kind of got this nailed down here. So I think our best chances to make progress in understanding spawning behavior is kind of turn the cameras away from that act and start to figure out what they're doing when they're not on the reef, what happens when the males arrive a month in advance, those kind of things. So I'll just end here by kind of giving a few concluding thoughts. These are kind of my overall impressions while I was doing these reviews. The first is that lake char are very diverse in both behavior and habitat use across the range, and this has probably led to their ability to thrive in a variety of different habitats. We do tend to see that individuals repeat the same behaviors year after year, but they also seem to be very highly adaptive and can respond quickly to environmental changes. A big one is that reality seems to be more complicated than the current conceptual models suggest. And an example I wasn't able to talk about today, but we're seeing equally interesting things, is spawning habitat use, where we have a very simple habitat model that explains some spawning sites great, but it doesn't explain why some sites that fit the model aren't used. And more recently, we're starting to see more and more observations of them using habitats that don't fit the model at all. So I think this one is a big one, this idea that reality is more complicated than we think. But I think we're also seeing a trend where recent advances in technology, things like telemetry, stable isotopes, otolith microchemistry, genetics, all that kind of stuff, is really increasing our ability to fill these knowledge gaps and hopefully will lead to more complete conceptual models in the future. And that is it. Are there any questions? Yep.
I was wondering if anybody's tried to test if there is gamete competition or sperm competitions in a group. It seems to me that might be something that could go on with the number of males there.
Off the top of my head, I'm not aware of anything. I know that's been done in other species. And the suggestion is that the closer that the gametes are released to the eggs, the more likely they are to be fertilized. But I'm not aware of any of that occurring in Lake Charr. Sorry? Oh.
For this good talk, I just wanted to ask you if you were aware of the studies on magnetic fields being used for navigation and orientation in the Chinook salmon sea eggs.
I am aware of them, and I've had several discussions with David Noakes about this. And his opinion was that on the scale that these magnetic fields change, they likely are not involved in Lake Charr navigation. But it's possible that they have different sensitivities and stuff. That he's basing that all on what's known about the salmon. But I thought about it. There's no research, I think, into that in Lake Charr specifically. But it is an interesting question.
One more question.
Thank you for that very interesting talk. I was wondering, because of what you showed on the films, that the size distribution of the males didn't seem to be very large. What is it? Is it much larger than what we are seeing?
In the videos I showed you there, these are fairly newly developing systems based on hatchery restoration. So basically every fish that we touch looks the same and is about the same size. So, all right, thank you
so much.